Today we have Andrea Idili as a presenter, and for those who joined for the first time, this is seminars that we have from time to time, but it takes on Fridays, and we discuss recently published papers or ongoing research in quite friendly environment where some people might even practice presenting, but it's also an opportunity to have a contribution at the scientific video journal. So because the presenter can choose, and then we publish this talk on YouTube. And I would like to introduce the speaker for today, Andrea Idili. I met him at a conference in Roma, I think 4 years ago. Now he's an assistant professor at the Department of Chemical Science and Technologies at the University of Rome, Tor Vergata, where he originally received his PhD in analytical chemistry under the supervision of Professor Francesco Ricci in 2017. During his PhD, he visited for up to 14 months the research group headed by Professor Alexis Vallée-Bellisle at the University of Montreal in Canada for external stay. After he defended PhD and a short postdoc at Francesco Ricci's lab, he did 2 other postdocs at University of California, Santa Barbara, USA, and at the Catalan Institute of Nanoscience and Nanotechnology, Spain. From 2021, he has returned to the University of Rome, Tor Vergata, as a Marie Skłodowska-Curie Research Fellow with the project Entropic DNA Sensors. His current research focuses on the development of novel DNA-based electrochemical and optical sensors able to support the real-time continuous monitoring of diagnostically relevant molecules, both in vitro and in vivo. The importance of his research is highlighted in more than 32 peer-reviewed papers published in high-impact factor journals, 1 patent, and 2 book chapters. He has received several international or national awards and fellowships in recognition of his work. I will not name them because it's a very long list. And the topic of today's seminar is in vivo measurements using electrochemical aptamer-based sensors. And I'm looking forward to hear it from you, Andrea.
Thank you, Antonio. So just to check to see if I can share properly and you can see nicely the presentation. Can you see?
Yes. Well, we see the, you know, with your notes.
Exactly. So I was okay. So let me do this. Sorry, I need to do this because I have a problem with the second monitor. Okay. So let's see. Okay. 1 second. Okay. Where is Zoom? Isn't it in okay. So you should be able to see, right?
Mm-hmm.
Correct, Antonio, can you confirm me?
Yeah.
Okay, so
Go
cool.
ahead.
I'm going to put this here. Okay, so before to start, I want to thank you for your, kindness and for this kind of invitation, because I'm super happy finally to share and describe my research activity. Today, I'm going to speak about on in vivo measurement performed using electrochemical aptamer-based sensors. So before to start to show you my research data and what I collected, I want to give you like a brief introduction, just to think how biosensor that can perform measurements directly in vivo can impact the current and the future of the clinical approaches. So now I want to push you to think, like to imagine in the next future where doctor could use implantable sensor on their patient. These implantable sensors can exploit a sensing technology that allow to perform real-time measurement directly in the bloodstream of the patient and can detect, in principle, any arbitrary molecules, for example, drugs, biomarkers such as antibody or nucleic acids or proteins or metabolites. Then the sensor is designed and engineered to send in real time all this clinical data directly to the doctor. So the doctor, keeping in his or her hand his or her mobile phone, can check and monitor in real time how for example, the drugs levels in the blood are changing over the time, or a specific biomarker. This could allow to the doctor to understand what is, for example, the effectiveness of a drug treatment, or what is the disease or health status of a patient, or in the scenario of an infected disease, reduce the time between the diagnosis and the starting of the drug treatment. So this could be the future, and this will allow to reach personalized medicine. But how can impact this technology, this in vivo biosensor, the current clinical approaches? So the ability of a sensing technology able to perform real-time in vivo measurement directly in the body of a patient, this could allow to achieve pharmacokinetic measurement and therapeutic drug monitoring with higher accuracy and precision. So pharmacokinetic studies are crucial for clinicians because they are fundamental to calculate the correct dose of drug. Here, I'm showing you the classic pharmacokinetic profile. So this pharmacokinetic profile is the plasma drug concentration plotted over the time. So, and here we can observe the three distinct phases of pharmacokinetic. Basically, this graph is going to tell us what the body does to the drug. So here we can see the classic absorption phase. So at T0, the drug will be injected. Here is for example, for an oral administration of a drug, there will be the absorption. Then when the drug plasma concentration will reach a maximum concentration here to Cmax, will start the distribution of the drug. So the drug starts to diffuse from the circulatory system to the peripheral tissue. Then we'll start the third phase, which is called excretion phase, which describes the elimination of the drug from the body. So plotting and creating this profile is possible to estimate important information. For example, what is the therapeutic range? What is this window where it's possible to understand what is the minimum effective concentration of the drug? So what is the minimum concentration to induce a therapeutic effect? And on the contrary, what is the maximum toxic concentration? So is the concentration that overcoming this concentration, you start to induce a toxic effect in the patient, so you can affect his or her health status. So plotting this PK profile, we can achieve pharmacokinetic parameters that are crucial, like the area under the curve. And this pharmacokinetic parameter, together the age, the sex, and the body surface area, are used to calculate the correct or the right dose of the drug. But what happened that the current approach that is used to obtain and to collect these pharmacokinetic studies, they display two major drawbacks that reduce the precision of the different drug treatment. So at date, this is the current approach to generate these pharmacokinetic studies. Normally, blood samples are taken from the patient. Then these are shipped to a specific laboratory, and the current analytical approach to estimate this plasma drug concentration are based on multi-step basis batch processes like HPLC or mass spectrometry or immunoassay. So because they rely on different steps and they have to treat the biological sample, the time resolution of this pharmacokinetic profile is very low. And this affects the precision how we can calculate, estimate this pharmacokinetic parameter. To better highlight and describe this problem, I show you two different pharmacokinetic studies. So on the top, you can see the pharmacokinetic study for a chemotherapeutic agent, irinotecan. So you can see for a pharmacokinetic of almost 8 hours, we can observe only 5 points, and the time resolution of these two different pharmacokinetic are multi-hour. The second pharmacokinetic is for the metabolite phenylalanine. Here you can see that over almost 3 hours of experiment, we can observe 8 or 9 points corresponding to the phenylalanine concentration in the blood. And at the same time, here the time resolution is almost 20-30 minutes. Okay, so fitting this data normally with a two-compartment model, we can obtain this pharmacokinetic data, but due to this low time resolution and low precision, this strongly affects this pharmacokinetic data. And the second major drawbacks is that each point represents the average between 4 and 6 animals. So this study are not performed on the single animal. So this cannot help to predict the variability in the drug response in the animal and then among the patient. And this is a big problem, especially for chemotherapeutic drug, because the therapeutic window is very narrow. So there is a higher chance to prescribe a wrong drug treatment. One of the like problem for clinicians is that they normally give those regimen that induced underdose instead of the overdose. So because it's due to this narrow therapeutic window. So the second major goal that we can achieve exploiting biosensor that can perform measurement directly in vivo is the holy grail of drug developers, is the feedback controlled drug delivery. This is an old concept, is an old idea in the medicine, but basically a biosensor that allow the high frequency real-time measurement in vivo of a specific drug could be coupled with a PID controller, and this could improve the precision how the drug could be administered, specifically the output of the sensor. This means its continuous measurement and estimation of the drug concentration in the blood can be used by this controller that can control in turn the infusion pump and induce microinjection in order to keep the concentration of the drug in the body constant and within the therapeutic range. This not only could improve the efficacy of the drug treatment, but could help doctor to reduce medical errors. In this graph, published by the Washington Post 2 years ago, is described that the third cause of death in the United States are medical errors. Now, in this huge statistic, there are different kinds of medical errors, but among these, there will be also wrong medical prescription about the drug dosage. So using this technology could help doctor to reduce the number of this number and help to improve the drug efficacy of the treatment. So let's recapitulate what are the 4 major benefits exploiting real-time in vivo measurement. First, we can improve the precision and the relevance in the pharmacokinetic and metabolic measurements. Second, we can achieve high-resolution patient-specific pharmacokinetic. We can achieve continuous monitoring, which is very important to achieve the high-precision dosing of the drug, of serum drug levels and health markers. And finally, feedback controlled drug delivery. Now, but to develop a sensor that is able to perform this measurement directly in vivo, we have to overcome 4 technical challenges. The first challenge is that the sensor must work continuously and must be reversible. Must work continuously, this means that has not rely on multi-step batch processes. So it can measure continuously with high frequency. And second, has to be reversible. This means that the receptor bound on the surface has to bind and release the target in a reversible way. So this will allow to the sensor to follow the increase the degrees of the target in the body. So second, the sensor must be reagentless. This means that we cannot add any exogenous reagents, but we have to use only the molecular species present in the organism. Second, third, must be selective. So the sensor must detect specifically the selected target in order to work directly in the bodily fluids. And the last challenge must be stable. This means that after we implant the sensor has to work in vivo per hour or per day, depending the specific application. Now, nature has already solved this problem, how to perform sensing in a highly complex background. And to give you an example of the highly complex background here, you can see these TEM images of nearly of the 2000 that describe the cellular environment. So nature has evolved as chemoreceptor biomolecular switches. These biomolecular switches are, for example, receptors that are able to bind their target through a conformational change. To better describe this idea, I show you here this natural receptor, which is the tyrosine kinase, that in presence of the insulin and after the binding, this binding induces a conformational change in the receptor, and this new state trigger a specific biological process. Here, for example, induce the opening of a channel for the entrance of glucose inside the cell. So the same signal transduction mechanism where the binding induce a conformational change and the conformational change induce or trigger some activity can be exploited, and we can use and we can exploit this signal transduction mechanism exploiting the programmability of the DNA. Specifically, it's possible to selectively program specific DNA single strand to bind non-DNA target with high affinity. And these single strand target are called aptamers. So the idea that was developed and we developed in the Plasco group was to exploit aptamer and engineer this aptamer to induce a binding-induced conformational change in order to use this nucleic acid receptor as a recognition element to build or engineer a biosensor. And these are the electrochemical aptamer-based sensor. So basically, we use a single DNA aptamer strand, and we modify this strand with that thiol group and a methylene blue as redox tag label. So the thiol group allow the attachment on the gold surface of the aptamer sequence. At the same time, the presence of the redox tag methylene blue on the other ends of the DNA sequence allow to track on the surface the conformational change of the aptamer. Specifically, in absence of the target, the methylene blue will be free to collide with the gold electrosurface, displaying a specific electron transfer rate that can be collected and tracked using square wave voltammetry. So in absence of the target, we can collect our voltammetric signal. Then the presence of the target will be recognized through the binding by the aptamer, which will induce in turn its conformational change. So in the presence of the target, the methylene blue will be closer to the electrode. This will increase the its electron transfer rate, and collected its voltammetric signal will observe an increase in the peak current. And this difference, the difference in the peak current, is correlated with the concentration of the target in solution. So using this platform, in the past, we demonstrate that this platform can support the continuous and real-time measurements of drug levels directly in the body of a living animal. So today, as first example, I'm going to show you how I developed a electrochemical Aptamer-based sensor to detect in vivo the aromatic amino acid phenylalanine. Specifically, the aptamer sequence, I selected this aptamer sequence because it was previously published and was present in literature. So the structure of the aptamer is a stem loop structure, where this stem is part of the primary region. Instead, this yellow portion is the binding pocket. So as working electrode, we use this gold wire electrode where its diameter is not 100 but 200 micrometer, and the exposed area, working area, is of 3 millimeter. So we functionalize the gold electrode surface of this gold wire electrode with the aptamer, and we passivate the surface with the mercaptohexanol. So after that, we fabricate the sensor. We tested our electrochemical Aptamer-based sensor in presence of the target. Specifically, we challenge the sensor in presence of increasing concentration of phenylalanine. As you can see, the sensor can respond to the target in its physiological relevant range. So here you can see that I collected the signal using two different square wave frequency. Why? Because the signal gain, the signal of this sensor are strongly affected by the square wave frequency used to collect the voltammetric signal. So for example, using high frequency at 300 Hz, we can observe a signal on behavior of the sensor. On the contrary, at 10 Hz, we can observe a signal of behavior. I will explain later why we can observe this different behavior. So after that, we characterize the sensor, and we can see that its useful dynamic range fall in the physiological relevant range of the phenylalanine. We characterize the specificity of the sensor. So again, to do this specificity test, we challenge the sensor in presence of the increasing concentration of the two aromatic amino acid tryptophan and tyrosine against a solution of 5 different amino acids, specifically glutamine, histidine, arginine, alanine, and proline, and then against the 2 metabolic derivate of phenylalanine in the body, the phenylpyruvic acid and the N-acetyl phenylalanine. So as you can see here, no one of the interferent can induce a detectable signal change. So the sensor results very specific. Then I characterize the binding kinetics. This binding kinetics is very important because is going to describe what is the time resolution of the sensor, and if the sensor can track metabolic change in the body. So to understand the binding kinetics, I collected the signal of the sensor for 5 minutes. after I achieved a stable baseline, I added in the solution 4 different concentrations of phenylalanine. And as you can see, the sensor promptly responds to the target and can reach the expected signal change in seconds. Then, to demonstrate that the sensor is fully reversible, I move the sensor from the solution containing working buffer and these 4 different concentrations of phenylalanine, and I move in a fresh solution of working buffer. And as you can see, the sensor promptly responds in this new environment, and the signal dropped down, reaching the original baseline value. So the sensor results specific and with a high temporal resolution, a second temporal resolution. Then the last step is to test the sensor in vivo and understand if this sensor is stable. So now I'm going to show you what happened in terms of the signal when we implant the sensor and we start to collect the voltammetric signal using square wave voltammetry. Here, we collected the signal using the 2 different square wave frequency, 300 and 1000 Hz. Of course, that if you change the frequency of the square wave, change also the value of the current. So after we implant and we start to collect the signal, we can see this drift of the signal, but the sensor can promptly respond to different intravenous injection of the phenylalanine. So it's clear that the sensor can respond to the target, but this drift observed at the 2 different frequencies does not allow to convert the signal in a concentration of phenylalanine. So this signal drift seems affect the performance of the sensor. So to solve this problem and to correct this signal, we apply this approach, which is called kinetic differential measurement. This is an engineering approach. So, and it's based on 2 different assumptions. The first assumption is based on KDM exploit the square wave frequency dependence of EB signaling. This means, as I told you in the first slide, that changing the square wave frequency, we can change the signal behavior of our sensor. Why? Because changing the square wave frequency, it changes the rate of the rate of the sampling of the electrochemical signal. So using low square wave frequency, we are collecting the signal more from the unbound state, which display a lower electron transfer kinetics. So for this reason, here we observe a signal off, because this represents the reduction in the population of the aptamer from not bound state to bound state. On the contrary, increasing the frequency, we are going to increase the rate how we are going to sample the electrochemical signal, and we can collect more current from the bound state. And we observe a signal on because introducing more target will induce the aptamer to populate the bound state with a higher efficiency. So this signal of we are tracking the bound state that increases with the target. So here, at this frequency that should be around 50, basically at this frequency, the current that we collect in the presence and absence of the target is 0, because in this specific point, we are collecting the signal from both conformations, from the unbound and from the bound state. So the total is 0. The second assumption of the KDM is that the signal drift observed for the 2 frequency has to be the same. So if these 2 assumptions are true, we can apply this formula where the current collected from these 2 different square wave frequency is normalized, and the current from the signal behavior is subtracted to the current of the signal of behavior and is divided for the average. Using this formula is a kind of NOT gate. We can correct the drift and remove, we can correct and remove this drift, so we can convert the signal in KDM signal that which will be converted to the phenylalanine concentration. So let's see if this KDM is able to convert and remove the drift observed in the sensor. Before to test the sensor in vivo, we calibrate the sensor using KDM, so collect the signal at 310 Hz. But for the phenylalanine binding aptamer, for the phenylalanine binding sensor, we overcome this problem that we met during in the calibration of the sensor, because normally to calibrate the sensor we normally use at 100% blood. But what happened that in the blood is already there is already phenylalanine, and the concentration of phenylalanine in the blood is between 35 and 85 micromolar, and this concentration falls in the sensor's useful dynamic range. So what happened that we cannot obtain a complete titration curve that can be perfectly fitted with a Langmuir curve. So to overcome this problem, we use this approach. We build the first half of our titration curve using a buffer which mimics the pH, ionic strength, and the protein content and sugar content of the whole blood. So we use this new working buffer as a blood dilutant. So, and we use the blood as a stock solution of phenylalanine. So we start to add increasing volume of the blood, and we collected the electrochemical signal. So we did this until we reach a concentration of 70% blood and 30% buffer, because at this concentration we move the sensor from this solution and we place in a solution containing whole blood, and adding increasing concentration of phenylalanine, we completed and we built the second half of the titration curve, of the calibration curve. Again, here we collected the signal at 300 Hz and 10 Hz, and then we converted in KDM signal. So now we have also calibrated the sensor. Let's see what happened in vivo. So this is the setup that normally we use to perform in vivo measurement. So we matched the working God electrode with a platinum wire, which will be the counter electrode, and with a silver wire covered with a silver chloride film, which will be the reference electrode. So these three wires display the same diameter, which is smaller respect to the in vitro characterization, because it's 75 micrometer. And we selected these sides because these three wires can be encased and inserted inside a 22 gauge catheter. This 22 gauge catheter is almost 4.5 centimeter, and it is small enough that can be implanted inside the jugular vein of a rat. So for our experiment, we use rat as animal model, and generally we use rat of a size between 300 and 500 grams, because they display a jugular vein big enough that the catheter can fit inside. Specifically, we did two surgeries. In the first surgery, we open the chest of the rat and we take the jugular vein, we apply a little cut in order to insert the catheter containing the sensor, and then we push out, apply like a pressure, we push out the wire sensor in order to be fully in contact with the bloodstream of the rat. On the opposite side, on the opposite jugular vein, we install the infusion line. So through a syringe pump, we can inject directly in the circulatory system of the rat the phenylalanine. During this experiment, the rat is anesthetized with isoflurane because I want to highlight this, it's not feeling pain. So all this in vivo setting is designed to does not allow to the rat to suffer or feel pain. So after that, we implant the sensor, and the sensor is fully in contact with the bloodstream. We connect with the potentiostat, and we start to collect the electrochemical signal using 300 Hz and 10 Hz. And this is what we are going to obtain. So on the top are the continuous and real-time measurement of the phenylalanine in vivo, and below you see how KDM can fully correct and remove the drift. So here you can see the high frequency of our approach. Specifically, I want to highlight that each point display a delay of 12 seconds. So the time resolution is 12 seconds. And with this high time resolution, we can collect all the three different phases of a classic pharmacokinetic. So the absorption, that it almost is impossible for the other analytical approach to collect this phase, especially for intravenous injection. Then we can see the distribution and the elimination. Very important is that here we can observe how the sensor can respond to the three different injections of the phenylalanine. So the sensor works. Let's see if we can use this sensor as tool to estimate metabolic differences. So before to do this, we checked that the specificity is still the same. So we repeated the specificity test in vivo using a blank solution. So it's the same solution containing the drug, but without the drug, a solution containing the tyrosine and the tryptophan. As you can see, there is no response. And the sensor is highly specific. Then we use this to estimate metabolic differences between the animal. So we select two rats to fasten the rat. This means that we didn't give food, we didn't feed the animal for more than 10 hours. Then we performed these two experiment, and we injected intravenously in the rats 100 mg/kg. We selected this amount of phenylalanine because it's the same amount that is performed in the phenylalanine test for people that suffer of PKU. So here, after that, we achieved a stable baseline. After 20 minutes, we inject the phenylalanine intravenously. And as you can see, again, the high time resolution allow us to track the absorption, the distribution, and the elimination. Then we use other two rats, but this time we use two not faster rats. This means that we fed the rat all the time. So as you can see, we repeated the same exactly experiment. The difference between these two animals respect to the previous one is the concentration of the phenylalanine post intravenous injection. Specifically for this non-fasted rat, we observe this baseline value is higher 50% more respect to the original value of the baseline. And this difference, we think that this difference is depending on this metabolic status. And also the high frequency measurement that we can achieve with this sensor allow us to estimate pharmacokinetic parameter with higher precision. So that give us also statistical meaning to this data. To better highlight this, I show you how we fit and we analyze this pharmacokinetics. So normally one of the most model used especially for the phenylalanine, that can be described by this two-compartment model when the phenylalanine dose is injected directly in the circulatory system, so it's totally available to the body. This the phenylalanine concentration start to change because it start to diffuse, no, and will be in equilibrium with the peripheral tissue. At the same time, it start to be eliminated. So fitting this pharmacokinetic with this two exponential fit model, we can achieve pharmacokinetic parameter with a very high precision, because here I described each parameter with a 95% of the confidence intervals. So here you can see the amplitude of the distribution and the elimination phase and their half-life. So here, comparing the data, we can see that they are very similar because these are all healthy rats, and due to the high time frequency of the sensor, we can estimate with high precision. And instead, if we are going to use on hold pharmacokinetic performed using HPLC, here we can see that after, over 2 hours of experiment, we have 1, 2, 3, 4, 5, 6, 7 points. So if we try to analyze these 7 points using a double exponential fit, as you can see here, and describing the error as the 90% of confidence interval, here we are going to achieve an error that is higher than the same value. So we cannot use a double exponential fit. So we are forced to use a singular exponential fit to fit this data and to obtain some pharmacokinetic parameter that has statistical meaning. So here you can see that almost the majority of these pharmacokinetic studies, all the operators are forced in the majority of the cases to use a different model to fit the data. And this leads to decrease the precision of this parameter. So now, really briefly, I'm going to show you, I'm going to show you, I don't know what happened. Sorry. I don't know. I have this, okay. I'm going to show you that one of the main advantage of this technique, that of this platform, that is generalizable. So I'm going to show you that only changing the piece of the DNA used, so changing only the aptamer used, we can detect another clinical relevant target. So I'm going to show you here how we can detect a chemotherapeutic agent, very important, which is we selected irinotecan. So to detect this therapeutic drug, this chemotherapeutic drug irinotecan, I selected an aptamer that was previously published and was already present in literature. The main difference between the previous sensor is in the aptamer. It's very important to note the different structure, because I want to highlight this, that probably I didn't do before, that the magnitude of the conformational change of the aptamer will affect its signal gain, so its analytical performance. So here we select this aptamer where the stem portion is part of the primary region used for the selection of the aptamer. Instead, here the binding pocket is a G quadruplex motif. So I use this aptamer. Again, I modified with the methylene blue and the thiol group, and then I fabricate the sensor. Then I tested in the presence of the target. So as you can see here, I normalize here. I show you this graph as normalized current. As you can see here, the sensor promptly responds to the target, and the clinical relevant range for irinotecan is between high nanomolar until 20-30 micromolar. So the sensor fall in the clinical relevant range of the drug. Then After that, we tested and characterized the dynamic range of the sensor in vitro in the working buffer. We test in the blood. As you can see, the signal gain decreased drastically. So to improve the analytical performance of the sensor, I re-engineered the aptamer on the surface, specifically to induce a more unfolded and open conformation on the surface. I start to destabilize the aptamer using two different approaches. I introduce a mismatch in the stem, then I start to truncate, deleting base pairs from the stem. I did this because the stem is not part of the binding pocket, so eliminating base pair from this portion should not affect the binding activity of the aptamer. So using these two different approaches, I generated 6 new variants, and then I tested in buffer. So as you can see, especially CA28 display a signal gain of more than 400%. So this re-engineering approach was successfully. Then I tested in blood, and again, in blood, we can increase the signal gain. So we selected as new variant, so as new aptamer variant as sensor, the variant CA28, which is this one. So again, before to test the sensor in vivo, I tested the binding kinetics, and again, the sensor can detect in seconds can reveal the presence, can detect the presence of the target in buffer. And again, the sensor is fully reversible because in seconds can reach the original current as in the baseline value. Then I calibrated the sensor here for this drug. It was easier because it's not present, it's not already present in our body. So I used blood and as you can see here, the main difference is that this drug is display a high affinity for the protein. So if you see the calibration curve, the affinity in blood is lower because the majority a fraction of the drug starts to be bound to the blood's protein. So, and they act a kind of sequester. So after this, we implant the sensor in the jugular vein of our rat, and we start to continuously measure the presence of irinotecan. Here, because this sensor does not display a signal of behavior, but display only a square wave frequency, which is non-responsive. This means that it's flat, does not give a signal of behavior, allow us to detect irinotecan. So we are still using two different square wave frequency here, 120 Hz and 10 Hz, to correct the drift. But here the frequency at 10 Hz does not display a signal of behavior, but this allows us anyway to detect irinotecan. Here you can see that we can correct the drift and we can detect irinotecan in its clinical relevant range. Then again, we tested the sensor with 3 different rats. So again, here I want to highlight that the high frequency of the sensor allowed to detect all the 3 phases, so absorption, distribution, and elimination. And the most important thing is that we can achieve single pharmacokinetic from the single animal. So these data are not an average between animals. Fitting the data, again, we can achieve pharmacokinetic values with high precision. Again, here we have the different phase, and for irinotecan, this chemotherapeutic drug display high variability, so patients respond to this drug differently, especially for the elimination phase. As you can see here, for the distribution, basically we observe the same value. Instead, for the for the elimination phase, and as you can see also the clearance is like the rate how we can eliminate drug is very different. So these data are well described, but what is already reported in the literature. And to demonstrate the versatility of our approach, we show that we can achieve this elimination phase with higher precision. How? We start the experiment in a rat, starting the infusion of the drug until we reach a specific, and we can say we saturated the animal with the drug. Then we stop the infusion and we start to collect the signal in order to describe the excretion phase, so we can achieve a longer excretion phase, elimination phase. So fitting this data, we can achieve with really high precision all the half-life of the elimination and the clearance of the drug. These are very important pharmacokinetic parameter, especially for this drug that display a high variability. So, in conclusion, we demonstrate that AB sensors are a platform technology that support the high-frequency real-time measurement of specific molecules, and this can happen irrespective to their chemical reactivity, and this works in situ in the living body of animals. Then the sensor's time resolution, I want to highlight seconds, enable the high-precision determination of physiological phases that are too rapid to be captured using traditional approach, which are laboratory-based. Then the resulting measurement define the pharmacokinetic of irinotecan and phenylalanine in individual animals, providing an unprecedented high-precision view of drug inter-subject pharmacokinetic variability. Then the sensor produce sufficient numbers of qualitative data to characterize the disposition kinetics of phenylalanine with good statistical significance, thus providing an unprecedented tools with which to study inter-subject metabolic variability. AB derivative measurement, finally, could also support a new paradigm for personalized drug delivery. Specifically, we have recently used the real-time concentration information provided by the AB sensor to inform closed-loop feedback control drug delivery. And this is, I want to finish my talk with this. This was recently published by Nets, which is now a professor at Johns Hopkins University, to demonstrate that EB sensor can be fully applied to achieve feedback control drug delivery, solving the problem for drug developers how to give a specific drug dose that works for everyone, where the output, the estimation of the drug performed by the AB sensor can be coupled with a PID controller, which can control the pump and can induce the microinjection, so high precision dosing of the drug in order to keep constant the concentration of these antibiotics, which is thermomycins, in this narrow therapeutic range for hours. And with this, I want to thank you for your attention, and I want to thank, of course, Professor Kevin Plaxton and Professor Todd Keeping, the University of California, Santa Barbara, also my previous supervisor, Professor Albert Mercoci, and now Professor Francesco Ricci, and of course, European Research Council, to give me the opportunity to come back in Rome through the Marie Curie individual fellowship, and thanks to the entropic DNA sensor. And thanks again to all of you for your attention.